Hello, everyone. My name is Alexandria Williams. I'm a PhD student in the chemical engineering department here at UML. I will be covering a new yeast biomanufacturing platform with a closed carbon cycle and how it will impact the biomanufacturing industry today. The biotechnology industry uses synthetic biology and metabolic engineering to induce cells to convert renewable feedstocks like glucose from plants to chemicals and medications. However, about 30 and 80% of the carbon in glucose feedstocks is wasted as carbon dioxide. To contextualize this problem, public bio-based pharma and tech companies release about 227 million tons of carbon dioxide annually, contributing negatively to global warming. While there are efforts going towards the reduction of CO2 emissions through better scrubbing equipment, there must be a significant change made to how this carbon dioxide is processed and used in industry. If instead we can harvest the waste CO2 and convert it to high energy chemicals, a new biomanufacturing platform with minimal carbon loss could be established, helping achieve carbon neutral goals to combat climate change. Advances in electrochemical catalysis have demonstrated significant success in converting CO2 into C1 and C2 chemicals that have a higher usability in the cells, such as methanol, formate, ethanol, and acetate, which can be used by cells to make product. However, some organisms lack or underexpress the enzymes necessary for consuming this mixed chemical feedstock. The ability to consume these chemicals can be enhanced through introducing DNA that codes for those correct enzymes, though. Before genetic alteration, the base experiments must be ran on organisms to see how well they can process C1 and C2 chemicals naturally. Here, we show that unconventional yeast, Yarrowia lipolytica, naturally consumes a mixed feedstock of ethanol, methanol, glucose, and acetate. Shown here as well, why lipolytica can process ethanol and acetate on their own. Despite this, our yeast still struggles to survive on the presence of solely methanol and formate as its feedstock. We plan to increase the metabolic efficiency by inserting customized DNA sequences that allow lipolytica to better ingest methanol and formate to increase cell growth and production. If we are successful, the biomanufacturing platform with a closed carbon cycle can be established where CO2 from fermentation is harvested, converted to C1 to C2 chemicals, and then fed back into bioreactors, allowing a product yield that is comparable and exceeds traditional growth from traditional glucose-fed cell production.